Okay, so welcome back to this next video in which we are discussing the adaptive immune response and immunosuppressant drugs. Okay, so we've now looked at the humoral adaptive immune response, and we now want to turn our attention to how we attack intracellular pathogens, which are within phagosomes. So we want to turn our attention to cell-mediated but in phagosomes. Okay, because this is very, very similar to the adaptive immune response for extracellular pathogens. So, what's going to start happening is that at the site of infection, you're going to get macrophages and dendritic cells, these professional antigen-presenting cells, and they will phagocytose the pathogen again. And obviously the pathogen will then try and live in the phagosome is the problem. This is the difference this time. These pathogens just, they laid down and died once they were inside the phagosome. Okay, whereas these ones are going to try and live within the phagosome. However, some phagocytes here and there, some dendritic cells or macrophages will have success. Some of them at least will take surface antigens off of the pathogen and will break them down into fragments, and therefore will present these fragments on MHC class 2. So even though the pathogen is an intracellular pathogen which lives within phagosomes, it's going to fail sometimes. Sometimes the cell will get the better of it, and some dendritic cells or macrophages will manage to break down their pathogen, or at least break down an antigen of the pathogen. Okay, they will then display these fragments on major histocompatibility class 2 molecules on the surface of the on the surface of the macrophage or dendritic cell. Again, because this antigen came from a phagosome, it's going to be put on major histocompatibility complex class 2, and that's the major difference between the cytoplasmic pathogens and the phagosomal pathogens, and that's what makes this phagosomal intracellular pathogen more like the extracellular one as far as the immune response is concerned. So what's then going to happen is this professional antigen-presenting cell will migrate to the lymph node, so it'll go in the lymph vessels up to a lymph node, and again it's going to search for a CD4 positive naive T cell which has on its surface a T cell receptor which is complementary for the antigen fragment that it is presenting. Okay, so again you'll get this same process occurring. It will search for a CD4 positive naive T cell with this complementary TCR. Then when it happens to find it, the MHC class 2 with the antigen fragment will form a complex with the T cell receptor, and then the CD4 will also bind to the MHC class 2, and that will deliver signal 1 to this naive CD4 positive T cell. Okay? Then you'll need signal 2, so the professional antigen presenting cell will then have to have the co-stimulatory molecules on its surface, so it will need CD40, and it will also need either B7.1 or B7.2, but it will have these because after all we have a pathogen infection, so it will have pathogen-associated molecular patterns which will have stimulated the pattern recognition receptors on the surface of the professional antigen-presenting cell, and therefore induced the expression of these co-stimulatory molecules CD40 and either B7.1 or B7.2. The CD40 will then bind to the CD40 ligand on the surface of this naive CD4 positive T cell, and the B7.1 or B7.2 will bind to the CD28 on the surface of this naive CD4 positive T cell. Okay, and that will trigger signal 2 within this naive CD4 positive T lymphocyte. So everything's the same so far. What will happen is it will diverge in a moment. Okay? Then the signal 1 and signal 2 will induce the expression not only of interleukin 2 but also the interleukin 2 receptor alpha component. The interleukin 2 will be secreted from the cell and will now want to act on interleukin 2 receptors on the surface of the cell. Now in order to have a complete interleukin 2 receptor, you need an interleukin-2 receptor alpha component, an interleukin-2 receptor beta component, and an interleukin-2 receptor gamma component. The beta and the gamma components are expressed constitutively on the surface of T cells. The alpha component is not, but we've just started producing the alpha component in this naive CD4 positive T lymphocyte that has received both signal 1 and signal 2. And therefore, it will trimerize, well, it will join with these beta and these gamma subunits and form this trimer, which is the interleukin-2 receptor. The interleukin-2 will then be able to bind to the interleukin-2 receptor, causing an autocrine signal, which then gives signal 3 to the CD4 positive naive T lymphocyte. Okay. This then causes this CD4 positive naive T lymphocyte to differentiate into a T helper naught cell, okay, and then it will proliferate into a whole population of T helper naught cells. And now is where it diverges. This interleukin-4, which causes the T helper naught cells to differentiate into T helper 2 cells, that will not come this time because the pathogen is not extracellular. It's intracellular within phagosomes. Okay, so you're not going to get the differentiation of these T helper naught cells into T helper 2 cells. So what happens instead? Well, basically, you are going to have this population of T helper naught cells. So let's just separate this all off, and I suppose I really should have started a new piece of paper, but never mind. We're doing cell-mediated phagosomal now. Okay, so we have a population of T helper naught cells, okay, and they are going to differentiate this time into what are known as T helper 1 cells. But what drives that differentiation into T helper 1 cells? Okay, so let's start off with our population of T helper naught cells, which are here, and again I'll draw 3 of these. Okay, so these are all T helper naught cells. Well, basically you somehow need to communicate to these T helper naught cells that they should become T helper 1 cells, and they become T helper 1 cells in response to these intracellular pathogens that are within phagosomes. Okay, so what we have over here at the site of infection, so where the bacterial infection actually is, you will have macrophages, so here is our macrophage, which will have phagocytosed this pathogen, thinking it was going to do a great job, but now the pathogen has got the better of it. It's winning this fight. It is just sitting within that phagosome and stopping phagolysosomal fusion. So the lysosomes aren't fusing with the phagosome, and the pathogen is just sitting in there quite happily, and it might well divide within there and, you know, do its thing. Okay, so this macrophage has a trick though. It can send out a cry for help, basically. If it has a pathogen that is pulling this trick where it is stopping phagolysosomal fusion, so for instance Mycobacteria species and Listeria species are very good at doing this, it can send out interleukin 12. Okay, this interleukin 12 will then go into the interstitial fluid, then into the lymphatic vessels, and it will drain to the lymph nodes, and then it will act on these T helper naught cells that at the moment are still within the lymph node, and it's going to cause their differentiation into T helper 1 cells. Okay, so we're now going to get a population of T helper 1 cells here. Okay, so these T helper 1 cells, at the moment, they're still in the lymph nodes. However, they're going to very quickly leave the lymph nodes, and they'll go temporarily into the blood, and then they'll circulate in the blood for a while, so they'll go into the bloodstream, and then they'll be recruited to the site of infection. So they'll be recruited by the endothelial cells of the blood vessels in the site of infection, and they'll end up in the interstitial fluid at the site of infection. Okay, so they end up in the interstitial fluid near these macrophages. Okay, so interstitial fluid. Okay, now what they're going to do is they're going to be right up near these macrophages here now, and they are going to produce a very powerful signaling molecule. Okay, So they're going to produce interferon gamma, IFN gamma for short. So the full name for this is interferon gamma. Okay, so the T helper 1 cells come to the site of infection and they release a huge amount of interferon gamma into the interstitial space, and this is now going to go back to the macrophages. And this is going to give the permission to these macrophages to use absolutely brutal methods. This is what this instruction is. It basically says, we don't care anymore. Use your brutal methods. It gives them the license to not bring back prisoners. Okay, so it's giving them new powers. I always think of Harry Potter here, where there's something about, I don't know which book it is, but I remember Sirius Black telling Harry about how Barty Crouch gave the Auras new powers. He gave them the power to kill rather than capture. That's what I would compare this to. This is Barty Crouch, and the macrophage is the aura, and it's hunting down this pathogen. And the interferon gamma is the command that you don't need to bring back a prisoner, you just can kill it. Okay? So it's a command that you can now use brutal and horrible, inhumane methods, if you like. Just destroy it. Before, the macrophage wasn't allowed to use all of its tricks, and now, once the interferon gamma has arrived, that gives it the permission to use brutal methods. Okay? So, the macrophage is now going to pull out a whole new toolbox of horrible tricks, and it will be able to destroy that pathogen, at least better than it was before. So this is how these T helper 1 cells are going to go back and help the macrophages, which are struggling to deal with this intracellular pathogen that is living within phagosomes. Okay, so that's the response to these intracellular pathogens within phagosomes. Now let's turn our attention to these intracellular pathogens which are just within the cytoplasm. Okay, and this is going to produce a totally different immune response, because this is going to involve not the CD4 positive T cells, but the CD8 positive T cells. However, the whole thing does again start off with a professional antigen presenting cell. Okay, such as a macrophage or a dendritic cell, which is currently at the site of infection. However, this time it does not phagocytose the pathogen. Instead, it gets infected by the pathogen itself. So here now is the pathogen that has come into the cytoplasm of this professional antigen presenting cell. And again, I'm drawing it as though it's a macrophage, although dendritic cells are actually better at doing this than macrophages. But I'm drawing it as a macrophage simply because macrophages are easier to draw than dendritic cells. Okay, so we have our intracellular pathogen here, and you can imagine that maybe it's a virus. Okay, now this pathogen will have a whole bunch of antigens associated with it, so proteins against which we can launch an immune response. So proteins that a human cell would never ever have. Okay, so let's have this as our little antigen here, as this blue box. Now, what will happen is the cell will end up with some of these antigens within its cytoplasm. Okay, and it might just break this antigen down and then display fragments of it on its surface on the major histocompatibility complex, class 1. Now you might think, well, what triggers it to do that? Well, basically all cells in your body do this continuously. All cells continuously take proteins within their cytoplasm, break them into fragments and put fragments on MHC class 2. Okay, so, sorry, not MHC class 2, that was awful. MHC class 1 this is. Major histocompatibility complex class 1. Okay, MHC class 1. So all cells continuously do this, and they end up with a huge number of self-proteins on their surface, but it doesn't matter. Okay, because you won't get a T cell response to that. What's going to happen now is that when you infect one of these professional antigen presenting cells with your intracellular pathogen, Okay, it ends up with the antigens of this intracellular pathogen within its cytoplasm, and it does what any cell would do. It breaks some of these antigens up and puts fragments of them on its surface bound to the MHC class 1. Okay, so here is this antigen fragment. So again, I know it's very confusing that I'm denoting the antigen fragment in the same way as I'm denoting the antigen, but never mind. Cope with it. Okay, so this is an antigen fragment. I suppose I should probably colour them in different colours or something. Okay, so this is the antigen fragment, and we'll have major histocompatibility complex class 1 in turquoise. Now, this is the important thing. If this actually is an antigen fragment of a non-self antigen, okay, i.e. if it's an antigen from a pathogen, then the pathogen will also have a whole bunch of pathogen-associated molecular patterns, and therefore those will activate pattern recognition receptors of this professional antigen-presenting cell. So the professional antigen-presenting cell will also have co-stimulatory molecules on its surface, which have been expressed because of the activation of the pattern recognition receptor. Okay, so let's say these are these co-stimulatory molecules, and luckily they're the same ones basically. So you'll have CD40 and B7.1 or B7.2. Okay, and then what's going to happen is this professional antigen presenting cell, which has its antigen fragment and it has its co-stimulatory molecules here. Here is CD40 in orange, and here is B7.1 or B7.2 in green. It's going to go in the lymph vessels again. It's going to go up to a lymph node, and then it's going to have to percolate through the cortex and the paracortex of that lymph node, and it's going to meet a whole host of naive T lymphocytes. Now this time it will be looking for a CD8 positive naive T lymphocyte because it's presenting an antigen fragment on MHC class 1, and CD8 binds to MHC class 1, not CD4. Okay, but apart from that, CD8 positive T lymphocytes are exactly the same as CD4 positive naive T lymphocytes in that you have a massive great number of them, and they will all have slightly different T cell receptors. And these T cell receptors will be complementary for different antigen fragments. So it's going to search and search and search for a CD8 positive naive T lymphocyte that has the perfect T cell receptor for this antigen fragment that it is presenting. So here is our MHC class 2 with its antigen fragment here. Let's say this is our CD8 positive naive T lymphocyte here. Okay, and we're going to form an immune synapse again. So this is a CD8 positive naive T cell. Okay, and it has the perfect T cell receptor. So our antigen presenting cell had to search and search and search for a CD8 positive naive T cell which had this perfect T cell receptor for the antigen fragment that the MHC class 1 has bound to it. Okay, so this here, this is our T cell receptor. Okay, here, this is the antigen fragment, so I'll put this as AG, which is, remember, shorthand for antigen. This is the antigen fragment, okay, and we'll colour in the antigen fragment in blue here. Okay, so this is the antigen fragment in blue, and then the MHC class 1 will colour in turquoise here. So this is MHC class 1, okay, which is presenting the antigen fragment on its surface. Okay, so this is MHC class 1, major histocompatibility complex class 1. Now, the CD8 positive naive T cell specifically has CD8 on its surface, which is also going to bind to the MHC class 1 here. So that's in orange. So this is CD8 in orange here. Okay, now, these two molecules, firstly the T cell receptor binding to the antigen fragment on the surface of the MHC class 1, and also the CD8 binding to the MHC class 1, these together are going to deliver signal 1 to the T cell. Okay, so it's the exact same sort of process, it's just we've replaced MHC class 1 with MHC class 2 and CD4 with CD8. So this delivers signal 1, okay, and then you'll have the co-stimulatory molecules here. So here's CD40 binding to CD40 ligand, and here is B7.1 or B7.2 binding to the CD28. Okay, so let's colour all of those in. So we'll have the CD40 in red here. Okay, we'll have CD40 ligand on the surface of the naive CD8 positive T cell in green. We'll have, what other colours have I got? Now I've used that one, yellow. Then we'll have B7.1 or B7.2 in yellow, and then we're going to have to duplicate, I think. We'll have the CD28 in pink here. Okay, so let's label these all up. This is CD40 ligand in green. This is CD40 here in red. This is B7.1 or B7.2 in yellow. Okay, on the antigen presenting cell, and I'll just give the antigen presenting cell a nucleus here. Okay, and then finally over here we've got the CD28 in pink. Okay, so CD40 ligand and CD28 binding to their co-stimulatory molecules on the surface of the professional antigen presenting cell will deliver what is known as signal 2 to this CD8 positive naive T cell, and these together then are now going to lead to the next step in the activation process for this CD8 positive naive T cell, and we'll go over that in the next video.